Okay, quiet folks. So yes, and then for proteins in eukaryotic system, they still go through the similar pathway, right? For translation, it's very, very similar. So a lot of changes are present in that initial part where transcription happens between prokaryote and eukaryote. But when it is time for translation, the basic translation process remains the same. Again, what's different is that once the protein is translated, there are further editing steps to make it active in many cases. You can get a lot more modifications in eukaryotic proteins than you see in prokaryotic proteins. Specifically, glycosylations, they don't do the same type of glycosylations prokaryotes. They don't have as many complexes of those. So there are things that eukaryotic proteins go through that the prokaryotic proteins may not go through. And that can directly affect their activity and their response in the system as well. So these changes that happen past protein being built are called post-translational modifications. And these post-translational modifications then lead to pool of functional proteins that can either do their work, or if they're not needed, they can be degraded, or they can be transported, whatever it is that they need to do. So here you have an example of how you have your newly synthesized protein polypeptide chain. It's going to bind to different cofactors to help it fold through those non-covalent interactions to get into the right folded space. They can have post-translational modifications such as phosphorylations, glycosylations, acetylations, methylations, sumoylations. There are lots of different things that can happen. And with the combination of all of those required changes, you can get your final actual functional protein, mature protein at the end of the day. So that's essentially what we will go through for that. And then let's go through today's topic. As well, since you guys seem to be good with that. So frankly, most of the stuff that we are going to have on the exam is going to be actually covered today and in this chapter. There's going to be limited stuff on the next one, but there are some important things that we will need. Okay, so today, for the next topic, we are going to look at how the gene expression is controlled and regulated. How does a cell know when to produce which transcript, when to translate what transcript to make functional proteins? We are going to look at it on different levels. So how transcription is regulated, how translation is regulated, how do we control that through post-transcriptional or post-translational controls, and then how does all of this help us generate specialized cell types? So that's essentially the basic of what it is that we're going to look at. So if we look at all our cells, right, all the cells in a multicellular organism, we are going to realize that they all contain the same genome, right? All of our cells and all the different body parts, they will have the same DNA, the same genome, the same sequences. Yet, at the end of the day, those cells look different, even having the same recipes inside them, having the same instruction book. And that comes down to what RNA and what proteins are getting expressed in each one of these different types of cells. And this process is very specific. It is controlled through specific differentiation factors and proteins that allow these cells to develop into what they are, right? So our differentiated cells contain the same genetic information. They have all the information to make any cell in the body, but at any given time, only some of these cells are, only some of these transcripts are getting built and produced into proteins and functional products. So there are several different experiments that helped us see and look at how these different systems develop and when these systems start to break apart so that you can no longer get a full organism, when cells get differentiated enough, for example. So here in the first example, they had cells from the skin of an adult frog, and they were looking at what they could get from there. And then they have an unfertilized frog egg. They went ahead and destroyed the nucleus of this particular egg using UV light specifically to that. And they took the nuclei from the skin cells and injected them into the egg itself. And they realized that they were able to get normal embryo and a tadpole. So they were able to develop it into a full frog. That means that a completely mature skin cell still contains all the information needed inside the nucleus to make a full organism. It just doesn't do it because of the environment that it's in, because of what's present in the cytoplasm as its differentiated form. However, if given the right environment, the same nuclear material could give you another organism as well. Similarly, here they have a carrot, a section from the carrot. They took a small section and they grew it in nutrient media. They were able to get proliferating cell mass, which they then separated out into individual cells, so kind of like a cell suspension in, again, a liquid medium. So this is a suspended culture rather than an attached culture. They took one of these cells and they were able to get clones from their young embryos and eventually a carrot. Again, showing that just a cell from a fully developed organism, just a somatic cell, just a normal cell, is also able to still give you a fully mature carrot given the right environment, the right system. And just like that, so this is how we develop the process of genetic cloning, right? And cloning organisms specifically. That's how Dolly the sheep was born, right? So here is another last example where you have epithelial cells from the oviduct, and these donor cells are placed into the anucleated eggs, just like we had up here. So they had the unfertilized egg cell, they removed the meiotic symbol and the spindle and the associated chromosomes, and they added the nuclei from these epithelial cells of the oviduct, and they were able to create a reconstructed zygote using the DNA from this particular nucleus. And that gave you the embryo that produced a calf. So any of these, all these different experiments give you proof towards this hypothesis that the nucleus contains all the information regardless of which cell type it comes from. So then obviously something's different to make certain parts of it silent in each of the cell types. And that comes in the form of those expression signals that they're receiving from the outside environment, along with the signals that are present within the cell itself. Each different type of cell is going to produce its own set of proteins depending on its environment and its own state. So there are some proteins that are common to all multicellular organisms. These are called, or all the cells inside an organism, and these are called housekeeping proteins. These are used for normal cell function, everyday functions that are needed in every single cell type. For example, all the cells need to be able to metabolize glucose, or make new amino acids, or make the ribosomes to have around so they can make new proteins. All those things still need to be made. But then there are other things that are going to be specialized proteins specific to the type of cell that you're looking at. And this can change based on the environmental signals that the cell receives, along with the differentiation state of that particular system. So gene expression can be regulated at many, many different parts of the process, not just at the very end or very beginning. So the first part is the transcriptional control. So this talks about 7 different places along this pathway, along the central dogma, where gene expression can be regulated. The very first control is at the transcriptional level, and this is usually directly related to the cell signaling that is being received from the outside environment through the plasma membrane into the cell. Once the transcriptional control is regulated and you have an RNA transcript, you have a second level of control in RNA processing. When and where the messenger RNA is going to be finely matured, when is that mRNA going to be transported and localized into the cytosol, when is that mRNA going to be translated or destroyed, and then once the protein is made, when is that protein going to just be hanging out versus when is it going to be degraded or when is it going to be activated. So you can have a protein made but not used, just hanging out in an inactive state, or it can be activated given the right environment, given the right signal, and do work. Or if it is no longer needed, it could be degraded and destroyed. So there are 7 levels of control throughout this process of transcription and translation of a particular protein. So we're going to kind of talk about the transcriptional regulation and then the translational regulation. And then finally, we will talk about overall signaling in later chapters when we get to them. So starting off with transcriptional regulation, there are 3 different things in transcriptional regulation that we want to care about, that we want to know about. The first is actually controlling the start of transcription. What controls which transcripts are going to be activated to be transcribed? So these are the transcriptional regulators, like transcription factors, the initiation factors, all those things that regulate, that bind to the promoter region, for example, and regulate which DNA sequences are going to be activated for transcription. The second is regulation of chromosome structure that can also control which sequences can be regulated and activated to begin with. So if you remember, we talked about 2 different types of chromatin. We talked about heterochromatin and euchromatin. Which one of those, by the way, if you remember, this is from this module, not this module, but this section. Which one of those 2 types of chromatin is more open for transcription? Euchromatin or heterochromatin? Yes, euchromatin is going to have a more open conformation. And what makes the heterochromatin more closed up? What's different about it? What are some things that happen to it? What are some features that make it more closed? So what controls whether it's going to be shut up or open? Yes? I thought I heard somebody begin to say something. A big part of it is methylation. Yes, you have those methylated portions. And what's it methylating? Is it methylating the DNA itself, or is it methylating something else?
The histone tails.
Yes, so it methylates the histone tails that hold it more tightly then, right? And then it gets all closed up and shut down. So even if there was a promoter region in that little space, it won't be activated because it won't be open for those regulatory molecules to bind to it. And then the third thing we'll have is the post-transcriptional processing and control. So that's when the messenger RNA is then processed and controlled in its ability to hang out or be translated. So talking about the first part, the transcriptional regulators. There's several different transcription regulators that can regulate either that can activate or inactivate DNA transcription from from specific sequences. There are proteins that contain specific DNA binding domains that that are recognized, right? That bind to the DNA at specific points, with specific sequences, and can do the function they need to do. There's some structural domains that you should know about that will help for the DNA to bind to these proteins. are alpha helices, zinc fingers, leucine zippers, and what we call homeodomains. So here you can see some of these domains present within the sequences and how they can help for the DNA to bind, right? So right here, you have these what we call zinc fingers. It uses zinc molecules as well within the sequence to help the DNA right here bind to these protein sequences. A lot of the DNA binding domains will contain alpha helices within them, and you'll see those as part of the structures. They help to kind of coil around the DNA's own helical structure and make a more stable union with it. Leucine zippers are another type of proteins. They'll usually work as dimers that kind of scissor around the DNA and bind to it and kind of clip around it. And then homeodomains are specific sequences that can interact with the DNA through non-covalent binding, through hydrogen bonding of its own, and make a stable conformation with that DNA through those as well. So that's kind of the structural part of those DNA binding proteins, those regulatory binding proteins, that they will many times contain alpha helices. They will have specific shapes that are known to be homeodomains, the leucine zippers, and zinc fingers. And then those proteins will then, when activated, get translocated into the nucleus, where they will bind to the sequences that they are made for. So many times you can have these sequences in tandem, or you can have just one particular sequence. So in this case, you have a regulatory sequence where the transcriptional regulator will come and bind to. If that transcription regulator has to work as a dimer, you will have a doublet there. So you'll have that sequence repeated twice, for example, allowing for two particular molecules to combine together to create that dimer as well. Here, as an example, you have a regulatory sequence for NANOG, which is a transcriptional activator. So each one of the transcriptional regulators will have its own sequence that it binds to. There may be some similarities. There are usually a lot of T's and A's involved, but they will have a particular sequence preference, a nucleotide preference on one of the strands that it is going to recognize and bind to in that regulatory system. So there are two different types of transcriptional regulators. You can have repressors or inducers. A repressor will definitely inhibit the activation of those genes. It will turn off and repress the transcription of the downstream gene, while an inducer will induce the activation of the genes that are downstream from that. They can bind directly to the DNA itself, or they can bind to the repressors as well to minimize their effect and activate gene expression. Same proteins can have completely different binding partners depending on which can have different effects, can act both as a repressor and as an activator depending on what binding partner it binds with. So here you have an example of MYC protein. MYC and MAX proteins. MYC is known for activating mitogenic signals and activating proliferation of cells. So when the MYC protein is activated and binds to the MAX in this particular EBOX domain, which is its regulatory sequence that it recognizes and binds to, it is going to lead to increased mitogenic signals and increased proliferation of cells. However, let's say that there was a change in the state of cell, where now you had activation of this MXT protein. MXT protein also binds to the exact same sequence, and it has a stronger binding than MYC to that sequence. And what it will do is that it will kind of make MYC dissociate and bind to the sequence itself. And this will lead to the repression of mitogenic signals, inhibiting further proliferation, and instead allow for differentiation of proteins to occur, or differentiation of that particular cell to occur. So the balance between the amount of MYC and the MXT protein will regulate whether the max regulator is acting as a proliferator or a repressor. It can work as both depending on which binding partner it's binding to. Bound to MYC, it's going to activate. Bound to MXT, it's going to inhibit. Prokaryotes, we talked about how they have one step of regulation by organizing their genes into operons. Operons is essentially, an operon is essentially a cluster of genes that all get transcribed and translated as a unit. Now, they will get transcribed definitely all together, but then depending on the environmental cues, they will be translated as needed through some kind of a transcriptional or translational switch. They're transcribed together when needed, and then they're translated together as well, giving you all the different proteins that are needed within that cluster of genes to do the work that they need to do. An example of that is the trip operon. This is to create tryptophan, right? Remember the tryptophan, we talked about how having just one codon to code for it actually helps in regulating when this protein, this amino acid is getting built and when it is available for the cell to use. There are a cluster of genes. There are 5 genes that are needed in order for tryptophan to be made properly, to synthesize tryptophan. These 5 genes are organized together in the trip operon under the regulation of a single promoter. That promoter sequence contains the tryptophan operator that is going to control when it's going to be activated and transcribed from this single promoter. Once it's activated, you will get a single mRNA molecule that will have the 5 different enzyme genes required for the production of these 5 different enzymes needed for tryptophan to be biosynthesized and made available to the cell. So these 5 proteins are then going to be made from the same mRNA transcript and then utilized for the work. Now let's look at just the promoter region of the tryptophan operon, the trip operon, and see how it is getting regulated, okay? So if you look at the promoter sequence, remember you have the -10 and -35 site. This is upstream of the start of transcription, where you have those promoter sequences that are recognized. Between those 2 sites is the operator sequence. So we are essentially just looking at this region right here. In a system, when there is a lot of tryptophan around in the system, the tryptophan binds to a protein complex that binds to the operator sequence when activated. This is the active trip repressor. It's only activated when there is tryptophan in the system to bind to it, and that allows it to know that there's plenty of tryptophan around. Tryptophan is not needed. It's going to be bound to the operator sequence within that system and repress binding of anything to promote the transcription of the gene. However, when there is less tryptophan in the system, the less it is, the less is available to bind to this repressor because all is getting utilized building more proteins. And so there will be less tryptophan around. That inactivates, if there's no tryptophan bound to the repressor, it inactivates the repressor, making it dissociated from the operator region, making that site available for RNA polymerase to bind to it. And now when RNA polymerase binds to it, it is going to walk down the street and start the transcription for the trip operon gene. So just by having a repressor that is controlled by the amount of tryptophan in the environment by allowing it to bind or not be available to bind, this is a very simple, elegant system that controls when you need more tryptophan, it gets activated and it gets transcribed, and then it can be translated. Now, any questions so far, by the way, before we look at a second level of control for this? So this was just at the transcriptional level. Okay. No questions so far? Okay. So now you have your mRNA, and now you want to also have another level of control as well. So here is another level of control for the tryptophan. So when you have the tryptophan in your system, you can have the transcripts. So here you have, sorry about that. This is your tRNA with the tryptophan codon in there, right? In this particular mRNA structure, there are several areas that can bind to each other and several areas that are going to get looped as open structure. This particular transcript can be present in two different forms, right? This is an mRNA that has multiple spots where tryptophan is needed. If there is tryptophan present, the ribosome can bind to it. It can add the tryptophan in the beginning, and it can go ahead and just keep working through it, no problem. And no activation of tryptophan operon is going to be needed. However, when there is no tryptophan present, or there's less tryptophan present, it is going to stall at that initial step where tryp codon is needed because there's no tRNA available with tryptophan. That's going to stall the ribosome, right? And it's not going to be able to create the full transcript. And instead, what you're going to end up with is this stalled structure with the stem loop, right? And this is going to, again, trigger the trip operon to be transcribed more, because this is another way that it is going to signal the cell that there's not enough tryptophan available in the system. So these are alternative stem loop structures that can happen inside the messenger RNA that can give you additional ways to regulate your system to activate or inactivate the transcription of a particular gene of interest. So activator proteins, just like in this case, you had the repressor with the tryptophan bound to it. You can also have activator proteins that regulate operon gene expression as well. In this case, your activator protein is going to get bound to the same promoter sites or the operator sites. And once they're bound, they are going to allow for RNA polymerase to come bind to it. They will find a place for the RNA polymerase to dock, activating downstream gene expression. So an example of that is another prokaryotic operon. This is the lac operon, and this is the last thing we'll do before we call it for the day. The lac operon is controlled by two different signals. It's controlled by a repressor that is going to repress the signaling or the transcription of the lac operon, and it is also controlled by an activator that is going to activate the production of the lac operon transcript. It's activated by both. So when you look at the LAC-Z gene, so here's your LAC-Z gene, you have the operator sequence, again, that's the start of transcription. And then you have the RNA polymerase binding site in the promoter region. This is your -10 and the -35. In this case, you see that there's the operator sequence is in front of the promoter region. And then behind the promoter region, there's a second regulatory sequence. And this is the cap binding site. This is where the cap activator binds. And that's where you're going to have further control of the system to allow for the polymerase to bind at the promoter region. So you have an activator, the promoter region, the operator, which is going to bind the repressor, and then finally you have the actual gene. When you have plenty of glucose and lactose around, the operon is going to be turned off because there's plenty of lactose, there's plenty of glucose. The cells are able to use the glucose as the preferential energy source. They don't need to worry about the lactose and its consumption or its production. They can just go on with the day. When there's plenty of glucose and not a lot of lactose, again, there's no need for operon to be activated. So not having any lactose around is going to allow for lac repressor to bind to the promoter, to the operator region, and keep the operon switched off here as well. However, when there is no glucose and no lactose, that is going to give the signal to the operon that it needs to be activated. And to do that, what happens is that when glucose is high, there is very little cyclic AMP around because the glucose is high, so it's getting broken down and you're getting plenty of ATP and you don't need to worry about it. But when glucose starts to get low, the cyclic AMP levels start to increase in the cells. When these levels are higher, they're going to be able to bind to the cap binding site, which is the activation site for this particular operon. And this is going to, the cap activator bound with the cyclic AMP is going to start to activate the process. However, the lac repressor, because there's no lactose around, is still bound to the lac operator region. And so the operon will still remain off and no transcript will be built. The only time the transcript will be built is when there's no glucose for the cell to utilize for ATP production. However, there is the secondary source of energy in the form of lactose present in the system. In this case, the lac repressor will not be bound to the lac operator region. The cyclic AMP and cap activator are already bound to the activation signal, allowing for the polymerase to finally bind to that site because nothing is blocking it from binding to the promoter region and starting the transcription of the lac operon gene. Okay, so this is a very, again, this is even more controlled operon than the tryptophan one, because it only gets activated when there's no glucose around as the preferential source of energy, and there's only lactose around that they can utilize. And so the only time they will activate that LAC-Z gene so that it can be utilizing those lactose is when it is needed for sure. This allows us to, we can actually use this system to help us examine when we are able to clone our genes properly, when we are able to examine by linking them to the promoter for the LAC-Z gene. And so then anytime our gene of interest is getting made, it's cloned properly, you're able to see the under the LAC promoter, you're able to see a color change and you're able to see that your genome is in fact transfected properly. So I'm going to stop here and we're going to continue from here next time in class and go through what we can along with the genome chapter. Okay. Any questions before we leave? So we talked about the prokaryotic gene expression control today. We will be finishing up the eukaryotic gene expression control next time, and that will lead us into the self-assembly and the differentiation portion of the chapter from next week. Okay? Guys are good? I'm being very quiet. Okay, see you next time.